here, and my lab, I'll present the BBQS grant that we have to develop synchronized neural and peripheral biomarker recordings in freely moving humans. I'll be moving the program to Duke University in a few months. And this slide, I pretty much feel like I don't have to mention to this crowd, but the goal of my lab is to understand, brain behavior relationships ultimately in real world settings, such as this one, very complex scene, but also how we can mentally transport ourselves back to these experiences through memory recall. And as was mentioned, traditional neuroscience, especially on the human side, has been very confined to constrained settings, laying still, lack of movement. And recently in this last, 5-10 years, we've developed these tools that enable us to get human participants out into the real world and moving and measuring brain and behavior relationships. So the way that my lab does this is we work with patients who have implanted electrodes. These are chronically implanted for disorders like epilepsy, PTSD, depression, others, and it's currently at around several thousand individuals who have these electrodes. You wouldn't know that a person has these, they're fully implanted, and here's an example of one patient who came in the lab who has one of these devices, and because it's fully implanted and shielded, we can have them maneuver through the environment without movement artifacts or other eye movement artifacts. We can track their movement using these on-body sensors, motion capture cameras, and we can also use things like virtual reality and augmented reality to simulate experiences with some control in the laboratory settings. In order to do this, we can also integrate wearable sensors that have been developing and advancing over this last decade, such as eye trackers, biometric recordings, and as I mentioned, on-body motion capture sensors. The technology that we created fits in a backpack that you see one participant wearing here as they maneuver through the room. On the bottom right, you can see the real-time recordings from 4 channels of electrodes implanted in the hippocampus that you can see here on the left side of the screen. And then we can, mark with millisecond precision and synchronization their position in the room, their speed, acceleration, their eye gaze, and their eye movements and other variables. We've used this technology to look at mostly studies of spatial navigation and memory, focused on the hippocampus, to see relationships with, like, for example, theta activity and walking speed when there's no memory demand. However, when there are memory demands, we see that that speed relationship is replaced by memory success. We've also found that relationships between eye movements, theta activity, and the ability to, sort of track the position of others, as well as distance to environmental boundaries, which can help us, sort of navigate our environment and remember certain target locations. And then more recently, we've been looking at more imagined experiences and showing that these relationships can also occur not just during real world movement navigation, but when we're imagining these experiences, these navigational experiences. We're also very interested in sharing the technologies with other labs, and we've done so to enable development of new treatments and understanding of disorders such as Parkinson's disease, post-traumatic stress disorder, obsessive compulsive disorder, binge eating disorder. And then we're also now recently trying to understand relationships during sleep and memory, as well as social interaction or observational learning. Now, this technology has really enabled my entire lab, and it's been a very productive time to be able to look at these kinds of questions, but there are always things that we can do to improve. So the current technology is, it's pretty bulky and heavy. It weighs about 9 pounds, and after a few hours of wearing this, it can get uncomfortable. And you definitely can't go out into the home setting, where there are no researchers, and expect that these participants can wear these types of technologies for long periods. So our current program is to try to optimize and minimize the weight and improve the wearability of the system. For example, here is one such area in which we will do this by enabling a very thin, flexible PCB coil to replace this heavy, bulky wand that has to be put on the head in order to wirelessly stream the data and also insert synchronization marks with peripheral biosensors. So this will, reduce the weight by half, the cabling, and also just make it increase the wearability for these studies. In parallel to this technology, we also have a system that can record single neuron activity in epilepsy monitoring units. So this is a totally different system that we've been developing over the last 10 years or so. And we've used it recently to record single unit activity from the hippocampus in a handful of patients while they're walking around in their hospital room. Now, this system, it was not used to, integrate these wearable sensors, many of these wearable sensors, although we started with looking at, eye movements and eye tracking. But it has a lot of potential to be improved to integrate these sensors. So we're adding an extra chip to integrate these biophysical sensors. And then lastly, we have collaborators in the program to develop a new biochemical smartwatch, which will enable sampling of cortisol and epinephrine in the sweat and enable integration with the previous Neurostack system, as well as the RNS system that I showed previously. And this will enable to look at, these critical hormones in human sweat that can, be very informative for studies of stress and anxiety
2
and
minutes.
other avenues. Okay. And that's really it. So just summarize here, we have our R61 stage, which has really two major goals, which is to optimize these neural recording platforms that can record single neuron and LFP activity in freely moving humans, and then integrate this with our current biophysical sensors and this newly developed biochemical smartwatch to enable peripheral and neural activity synchronization. And then hopefully, if all of this is successful, we can move into the R33 phase, which will enable us to do human testing during freely moving naturalistic behaviors under neutral and stress-related conditions. So hopefully this future where we can record these kinds of signals in freely moving humans in naturalistic settings at home is closer, with the development of these technologies to the point where we can eventually get here. So with that, I'll just thank the lab members and contributing alumni, some of which who are here today, as well as our clinical collaborators, industry collaborators, and funding. So thank you all for listening. And if you have any questions, I'm happy to take them.